This is part 3 of the chapter 3 lecture notes on important biological macromolecules. And we are on the very last group, the nucleic acids. And there are two types of nucleic acids. They are DNA and RNA. And the only difference in their name is that DNA stands for deoxyribonucleic acid, and RNA is just ribonucleic acid. And of course, the function of nucleic acids is for, they are our genetic material. They're responsible for coding for our traits. Now, nucleic acids are similar to proteins and carbohydrates in that nucleic acids are polymers that are made up of nucleotide monomers. The monomers of DNA and RNA are nucleotides. So we would say that DNA and RNA are polymers. Each nucleotide has three components. Each individual nucleotide has three components: a nitrogenous base, and that base can be one of four bases: adenine, thymine, cytosine, or guanine. A pentose sugar, which is either, if it's DNA, the sugar is deoxyribose, and if it's RNA, the sugar is ribose. And then a phosphate group, kind of like there was a phosphate group in the hydrophilic heads of the phospholipids. So this shows you the 5 different types of nucleotides. DNA is composed of combinations of adenine, cytosine, guanine, and thymine, whereas RNA is composed of the same first 3 nucleotides, adenine, cytosine, and guanine, but instead of thymine, RNA is composed of uracil. So different combinations of A's, C's, G's, and U's. You can see that the cytosine, thymine, and uracil are in a group or a class called pyrimidines because they are single ring structures. And the adenine and guanine are in a class called purines because they are double ring structures. So those are the nitrogenous bases, the pyrimidines and the purines. An entire nucleotide would be a phosphate, a sugar, and a base. So this would be an entire nucleotide here. The structure of DNA is, sometimes we call it a twisted ladder or a double helix. In other words, it's double-stranded. The phosphates and the sugars form the sides of the ladder, the backbone. And then the bases form the rungs of the ladder. So here and here, the steps of the ladder or the rungs are formed by the bases. So the bases are the steps or rungs of the ladder. And each step is composed of 2 bases. The way that you know which 2 bases have paired up is based on the complementary rule. A's can pair with T's and C's can pair with G's. So if you, for example, one of your DNA strands is A, C, G, T, then the complementary strand would be T, G, C, A, because the A's can pair with T's and the C's can pair with G's. RNA is only a single-stranded molecule, and it is composed of the bases A, G, C, and U. And with RNA, there are 3 different types. actually four major types of RNA. I'm sorry, I've always taught that it was three. There is messenger RNA or mRNA, there's ribosomal RNA, rRNA, and then there's transfer RNA or tRNA. And I have to be honest with you, I have never learned about microRNA, so that would be little I RNA. But anyway, that's news to me. We're learning this together, I guess. Every year it seems like there's something new in the textbooks with biology. So I didn't read ahead, so I apologize for that. But anyway, there's a fourth type of RNA called microRNA. All right, messenger RNA. The job of messenger RNA is that it carries the DNA code into the cytoplasm of the cell for a ribosome to use that code to make a protein. So the way that the process works is that the DNA, which is found in the nucleus, is so big that it can't leave the nucleus, but proteins are made outside the nucleus. So the DNA makes a copy of the code in the form of messenger RNA, which is a smaller molecule. And then that messenger RNA carries the code outside the nucleus to a ribosome, and it codes for a particular protein. So you've probably heard that DNA codes for our traits. Now you know DNA codes for specific proteins because we're made mostly of proteins. So the proteins are going to determine our traits, like our skin color and our hair color and our height, whether we have big bones or petite bones, that kind of thing. The shape of our face, our facial features, all those things are determined by proteins. So messenger RNA plays the role of carrying the DNA code into the cytoplasm so that it can code for a particular protein. Ribosomal RNA forms the ribosomes, and the ribosomes actually use the messenger RNA along with the transfer RNA to make proteins. So what we learn about ribosomes is that ribosomes synthesize proteins. And this is the reason all types of cells have ribosomes. Whether they're prokaryotic or eukaryotic, they do contain ribosomes. So even if a cell doesn't have a nucleus or membrane-bound organelles, it's going to have ribosomes because all cells have to make proteins. The transfer RNA is a very small molecule of RNA. It only contains 3 nucleotides, and it is designed to carry or transfer amino acids. Since proteins are made of amino acids, it transfers the correct amino acid to the place that it belongs in the protein. So it carries the amino acid to the ribosome in the correct order. And this kind of shows that process. We have a whole chapter on this. This part of the process is called translation. The first part where DNA makes codes for messenger RNA is called transcription. But what you can see here is a growing peptide chain, and all of these little individual round molecules, the yellow, the blue, the red, there should be a fourth color. Well, no, not necessarily. These are amino acids. I'm sorry. There's 20 different types of amino acids. But it's just showing you this chain of molecules is a chain of amino acids. In each individual round molecule is an amino acid. So the transfer RNA is this molecule. The messenger RNA is down here. And the ribosome is in the background. The ribosome has a small subunit that's a little darker gray, and then a large subunit that's a lighter color. But that is the ribosome. So the messenger RNA is attached to the ribosome, and the transfer RNA comes in and brings or carries an amino acid, which eventually is going to be linked with the growing peptide chain and to make it longer. And then eventually the transfer RNA is going to leave, and it can be used over and over again. So that process is called translation. Now for the one I didn't know, the microRNA is the smallest RNA molecules, and their role involves regulating gene expression by interfering with the expression of certain messenger RNA messages. Okay, so we're just going to say for now that they help to regulate gene expression. I'll give you an example of what this means. So you have skin cells, right? And your skin cells, you need them to do certain things. You need skin cells to protect your body. That's what your skin does. But you need your skin cells to contain certain pigments so that your skin tone matches, you know. Um, but anyway, your skin cells do play a different role in your body than, say, your muscle cells. You don't want your skin contracting all the time. You want to be able to move your skin by moving your muscles, but you want your muscles to contract. You don't want your skin to contract. But your skin cells contain a copy of every gene that you were born with. So some of those genes have to be turned off. All right, so in individual cells, what makes them different is they contain a copy of all the same genes, but some genes are turned on and some are turned off in cells. Okay, and this is a comparison, contrast of DNA and RNA, similarities and differences. The functions for DNA is that it carries genetic information. It contains the genetic code, and RNA is involved in protein synthesis. And microRNA is involved in gene regulation. That's not listed up here. The location for DNA is that it stays in the nucleus. The RNA is formed in the nucleus, but it leaves the nucleus and enters the cytoplasm of the cell, which is just the fluid part inside of the cell. The structure is that DNA is a double helix or double-stranded ladder, and RNA is single-stranded. The only time, it says usually, the only time RNA is double-stranded is that some viruses have double-stranded RNA for their genetic material. But viruses are not technically a living thing. But living things, including bacteria, which are the smallest living things, their RNA is single-stranded. The sugar in DNA is deoxyribose. In RNA, it's ribose. The pyrimidines are cytosine and thymine in DNA, and they are cytosine and uracil in RNA. But the purines are the same in both, adenine and guanine. And remember, the pyrimidines have a single ring structure, and the purines have a double ring structure. The central dogma of life, I've been explaining that to you all along, but the central dogma is what I drew that says information flows from DNA to messenger RNA, and then that is used to code for the protein. And the process where DNA codes for messenger RNA is called transcription, and when RNA helps to build a protein, that's called translation. And this is the central dogma of living things. All living things follow this process for making proteins. This is a comparison chart of the 4 basic macromolecules. We've got proteins, lipids, carbohydrates, and nucleic acids. Excuse me. The monomers of proteins are amino acids. Examples are enzymes, some hormones, some structural proteins, some are antibodies or immunoglobulins, some are for transport, like hemoglobin. There's numerous examples of proteins. And then let's skip down to carbohydrates. The monomers are monosaccharides, simple sugars, and they function primarily in energy storage. And the monomers of nucleic acids are called nucleotides. And then when you go back to lipids, the reason I skipped over that one is because lipids don't really contain monomers, but the molecules that make up a lipid are fatty acids and glycerol. And back to carbohydrates, one other thing. Carbohydrates and lipids both contain carbon, hydrogen, and oxygen. But carbohydrates tend to have a 1 to 2 to 1 ratio.